because she always makes me look not very smart. That's her talent. And so back in 2002, there was the United Leukodystrophy Foundation conference in DeKalb, Illinois. I don't know if anybody have ever heard of this city. It's like DeKalb, Illinois. It's 90 miles away from Chicago, a lot of cornfields. And for 20 years or so, we had annually this meeting there. And so all these people would fly in from Europe, meet each other there. And she came up and she presented a series of 7 patients. Actually, it went the other way around. There was a patient, and she said, oh, I know what this disease is, and it's a new disease. And so Dan were like, well, what is it about? So she said, well, I have now 7 patients who have this disorder. It's a brain disease that is affecting the brain, but it's also affecting the corticospinal tract, and it's affecting the dorsal column. And I want to call it LBSL. And I said, well, is it a mitochondrial disorder? Because there's this lactate elevation. And she said, no, we've looked at the mitochondria. It doesn't look like it. We haven't figured it out yet, but I'll let you know when I find it out. Now, she always knows what she's talking about, but she won't disclose it. So about 6 months later, her paper came out, and she actually included that one patient that was presented there. So the paper ended up having 8 patients with this disorder. And so this was the first description of this disease at that time point. And then a couple of years later, I guess it was maybe more a couple of years, she then identified the gene. She found out what the gene is. And so since then, we can now use this genetic test as a way to diagnose this disorder rather than using this MRI criteria. But the MRI criteria are really the reason this disease was identified in the first place. So one of the advantages of Professor Van der Knaap is that she gets all these MRIs from all over the world. She's a genius and established this database where every MRI is recorded with the areas that it's affected. And so she has thousands of MRIs, and she will say, oh, I see a patient who has this and this and this. How many do I have like that? And so that way she can nail it down, and she can basically discover new diseases. And then she goes on and discovers the gene. And so what is the anatomy of LBSL? And this is still the easy part. The talk is going to get a little harder. So we're going to talk briefly a little bit about neuroanatomy here. So as most of you already probably know, the brain has basically roughly 2 areas. You have the gray matter and you have the white matter. The gray matter is called gray because the neurons, the nerve cells are sitting there. And so it's this layer here on top of the surface of the brain called the cortex. The white matter is this area here that looks kind of pink, yellowish white. And the reason it's white is because it has a very high fat content. Why does it have a high fat content? When you zoom in here, what you will really see is that the nerve cell, let's see if this works. The nerve cell that's sitting here, this is the body of the nerve cell, has a cable that goes to connect to the other nerve cell. So here's a nerve cell, and there's a connection going to this side of the brain and then to the rest of the body. And these nerve cells are wrapped around by a sheet of fat that we call myelin. And you probably all know this. It's important to note that this process of myelin production mostly happens after birth. When a baby is born, there is very little myelin in the brain. And this process of developing the myelin sheet that wraps around the cells, that really happens within the first couple years of life. The myelin sheet is not produced by the nerve cells. That's important to note. So you have the nerve cell coming out, and then there is another cell type called the oligodendrocyte that goes in there and wraps this nerve cell with the myelin sheet. So the myelin sheet is produced by a cell type called oligodendrocytes, and then there is the neuron, which basically sends the signal. And the reason we have this fat layer is to insulate the neurons and increase the conduction speed. So when you have an abnormal myelin pattern, what will happen is that the conduction speed will be slower, so processing of information in the brain will happen slower. So the first thing that she used, Dr. Van der Knaap, to define the criteria is that these patients all had lesions in the brain in the cerebral white matter. The second thing that she said is that these patients all had lesions in the spinal cord tracts. And so let's talk about 2 spinal cord tracts that are part of this disease. One tract is the sensory pathway that takes the signal from our feet and from our fingers to the brain. there is different sensory signals. There's pain, there's touch, there's temperature, but there's also positional sense and vibration sense. Positional sense is extremely important. When we walk, the reason we don't have to constantly look at the floor is because our feet are constantly having these sensors that are measuring where we are in space. And so every single step we take, our feet automatically measure where we are in space by measuring our positional sense and sending that signal to the brain. Now, how is that signal sent to the brain? The signal is sent to the brain by these sensory fibers. So these fibers, these blue fibers, they are sitting in our toes and on the bottom of our feet, and they take the signal to the back of the spinal cord, and then from the spinal cord take the signal all the way up to the brainstem in the back of our head. They cross sites, and then they go to the surface of our brain where we actually feel it. So that's one of the sensory pathways that measures the positional sense and vibration sense. And these fibers go in the back of the spinal cord. So the back of the spinal cord here is called the dorsal column. It's a column and it's dorsal, so it's in the back. Then there is another pathway, and that's the pathway that when I want to move my fingers, I think here, hey, move your fingers. And there's a signal that's generated here on the surface of the brain by the nerve cells. And that signal goes down to the brainstem again. It crosses again to the other side, and then goes down on the side of the spinal cord, on the lateral aspect of the spinal cord. So this one is called the lateral corticospinal tract. And so Dr. van der Knaap's initial description was that this disease has to have 3 MRI criteria to raise your concern for LBSL. One is that you have changes in the white matter in the cerebral white matter. So you see here an MRI of a patient. This area that is red is the area here that's bright, and that's the abnormal signal here. On this MRI, a dark signal is good. The brighter the signal, the more abnormal you are. The other thing that she noted is that you have to have changes in the dorsal column, this sensory pathway, and in the corticospinal tract, in the lateral corticospinal tract. And so here is a picture of the spinal cord, and you can see here the dorsal column is bright, and the lateral corticospinal tracts are bright. So when you have these 3 criteria, you become very suspicious about this disease. But you should confirm it by doing genetic testing. And we're going to talk about the genetics a lot more, but just before we go there, let's think about this disease. So what forms does this disease have? And this is something that's evolving over time, and we're learning every single day. So this is not final yet. This will change the more we learn about this disease. The initial description of this disease was 7 or I guess 8 children. These were children that had the onset of symptoms usually after 1.5 years of age. So that would be what we call childhood form. But meanwhile, we have learned that there are people who are having a very early onset presentation, can be right at birth, or in the first few months of life, so infantile. And then there are individuals who don't have symptoms until adolescence, and some even until adulthood. And it's important to keep that in mind. There's also a couple reports suggesting that sometimes you may even have siblings in the same family, and one may have an earlier presentation than the other. But that is something that we're still trying to understand. So let's talk about these forms more. Everybody on board so far? Okay. So the childhood onset form. This is the form that we know most about. I personally have seen the most patients with this form. Ellie would fall under this category. And so these are kids who are initially normal development, cute as it gets. I think there is one thing abnormal in them. They all look cute. So I don't know what that's about, but there's something about it. And then usually start having issues first with balance. And it becomes notable as they become a little wobblier as they're walking. And usually it's an episodic decline. It is very important to note that this is a disease where it seems that the decline tends to happen in an episodic fashion. So it's not necessarily, there is some gradual slowing perhaps, but it's really an acute episode usually that triggers it. And it can be an infection with a virus, it can be dehydration, it can be a head trauma. And that's important to note because that's going to affect how we're going to take care of somebody who has this disease. And then there is some stiffness that can evolve in the legs, not in everybody, but some people develop that. And it tends to be more later, not in the first decade, more in the second or third decade. Some individuals, actually most individuals, are quite clumsy in their hands with the childhood onset form. They may have a tremor that's notable when they are trying to write or when they're trying to eat. In most of these individuals, as far as we know, cognitive function is completely intact. They don't have any intellectual impairments, they don't have any intellectual disability, but we haven't studied it in detail, and that's one of the things we would like to do going forward here with the studies that we're going to talk about tomorrow that we are proposing. There is only data available on 3 adults who have been studied more extensively with formal IQ testing. And it seems that in those 3 adults, there was some problem with processing information and working memory, not to a degree that would become notable. You would go ahead and be able to do your work. We obviously have a lawyer here in the room. We have another individual who's coming who's older, who is also a lawyer. But everybody has some deficits, right? I am inattentive. I'm quite inattentive, and some of the people in my lab think I'm worse than that. But everybody has issues. But these are subtle findings that we see. So there's no clear, obvious, loss of intellectual function that we have seen so far. There's one individual that we met a couple of years ago from Russia, and I'm not sure how much of her symptoms are related to this disease. She was normal as a child until her early 20s. She went on and got a PhD in economics in Russia and started having symptoms in her early 20s. She was very athletic prior to that. When we saw her, she was 33, and she had some stiffness and balance issues, able to walk without a cane, so no major motor disability. She had gotten quite depressed in the last 5 years, and when we saw her, she was cognitively quite slow. When you have depression, that might affect your cognitive function. So we weren't sure at the time when we saw her, is this cognitive slowing because of the disease, or is the cognitive slowing because of the depression? And so that's the only information we have so far. Unfortunately, she hasn't come back. She was from, not even Moscow. It was, I forgot the name of her city. It's somewhere in mid-central sort of Russia. And they came somehow one time. And they never came back. And the mom didn't have an email, and she didn't have an email. So I have no idea how they're doing today. So the next thing would be to talk about is maybe this adolescent or even adult onset forms. This is a form that we know very little about. But there are reports in the literature, and it appears that usually symptoms start in the teen years, and almost always they get misdiagnosed as having multiple sclerosis. And especially there's a form of multiple sclerosis called transverse myelitis, and that's almost always that's the diagnosis that they get until somebody comes and say, hold on a second, this doesn't really look like multiple sclerosis. And so in these individuals, it seems like it's a similar course like in a childhood form, but it just starts later, and it may be even slower in terms of disease progression. One thing to note here is that there's a handful of adults that we know of in literature, and a couple that we've seen here, who also developed a peripheral neuropathy. Peripheral neuropathy means that it's not just the brain and the nervous system, the central nervous system, but the periphery nerves, so the nerves that go from our cord to the muscles. So when you get peripheral neuropathy, you may get sensory issues, so you may get burning sensation, loss of sensation, tingling, numbness, and so that has been reported in some patients. And then, this is not an easy talk, but I think we need to talk about this. There's also now a lot of data emerging on the very severe form of this disease. And this is what we would call neonatal or early infantile form of this disease. These are individuals that are potentially symptomatic right at birth. They may already have a small head size at birth. They may have low muscle tone at birth. You saw the MRI I just showed you. When you look at this MRIs here, you get a sense how much more it affects the brain. So if you look at the MRI here, how much of the brain's white matter is white, most of it is white in these cases. So this is a more severe form of the disease, but we don't know a lot about the progression. And also, we don't know how the MRI actually correlates with the severity of the disease. We have had a couple individuals here who look clinically great. They look almost normal, and yet their MRIs look almost like this. And so there does not seem to be a good correlation between how severe the MRI looks like and how a patient looks like. And so all we can tell right now is that it looks like if you have the early neonatal form and the infantile form, you're more affected. It's low muscle tone, global delay, and some individuals also have seizures. And I think I just heard your story. I think that's probably the most pronounced one so far. So there is a study that has tried about 5-6 years ago to put together all the information that we have on a limited basis. And so when you look there, this was again done by Dr. Van de Knaap's group. When you sort of break it down in individuals who were less than 1.5 years of age at time of onset of the symptoms compared to those who were more than 1.5 years of age compared to the time of symptoms, you see a big difference. So those kids who are older than 1.5 years of age when the first symptoms start, as you can see here, this is 10 years, this is 20 years, this is 30 years from the day of symptom onset, okay? And so they do end up needing a walking gait at some point, usually after 10 to 15 years. About 60% are still able to walk without an aid, but 40% will need some form of an aid to be able to walk. On the other hand, if you look at the individuals who had the early onset form, you see that at least half of them very quickly within the first few years will need some walking assistance. And so when we say walking assistance here, that doesn't mean necessarily wheelchair. What this means is it could be a cane, it could be a walker, or a wheelchair. So when we look here again, this is another plot. When you see here, this is breaking down the patients in these different categories. So less than 2 years, 2 to 6 years, 6 to 12 years, 12 to 18 years, and then the adults. And so this is the time of onset again. So when did they have their very first symptom? And if you look at it that way, the black is the 2 individuals who passed away. So only in this neonatal severe form is where we have seen somebody actually passing away from this disease. But also in this category, you see the darker color means more disability. So the younger the onset of the disease, the more severe is the disease. The later the onset of the disease so far, as far as we know, the milder is this disease, which kind of makes sense. So now it's going to get a little complicated. I'm going to start talking about the gene, and I'm going to do my best. If you get 60% of it, that's great. Okay? If you get 100% of it, that's fantastic. I still don't understand it all myself. So just to be fair to everybody. All right. So yeah.
I was going to say, if there are words that pop up in this, or terms, or entire topics that you don't understand, either pass it to me or feel free to just write something up on the board, and we can try to make sure that we get that stuff documented. And we thought after the event, sometime in the next month or so, we could get some information out. But if something comes up that's just completely impossible to understand, again, make a note of it, and we'll get the information.
And another, I forgot to make this announcement, very important announcement. In your folders are all the bios, but more importantly are the presentations. So if you want to take notes, you've got it right there in front of you. Okay? Thanks, everybody.
Okay. So we're going to try to talk about the gene and what is the function of the gene as far as we know. And I'm going to start really simple. And I apologize if it's too simple for some of you. I just want to go really simple because this is very basic biology. So we have in our brain billions of cells, about 20 billion cells. In each cell, we have the center of the cell, which we call the nucleus. And in the center of the cell is where all our genes are. Well, almost all of our genes are. So we have about 24,000 genes. They are wrapped up and packaged in something called the chromatin material, and then they look like chromosomes. So I'm sure you have heard the word chromosome. So these are our chromosomes that have all the genetic material. So if you zoom in on the chromosome and you open up this chromosome, actually in each cell's DNA, if you think about it, if you open up the DNA, you can go from here to moon and back. So it's really long, okay? So 3 billion DNA letters. And so each DNA letter is basically a base, a nucleic acid base, and we have 4 of them. These are the 4 different colors. We don't need to go into that level of detail, but just so you know. So a gene, by definition, when we say a genetic disease, or if you say a gene means a fragment on the DNA that is having the coding information, or the information, the recipe, so to say, for the product that it's supposed to make. So genes are basically, and I think Becky might get into that in more detail, are like cookbooks. Each of them are like recipes for a product. The products are usually proteins. Proteins are basically the things that make up everything. You know, our cell borders, our cell organelles, our cell machineries, our cell engines, and so on are all made out of proteins. So proteins are sort of the biologic structure of everything. And so in order for the proteins to fold the right way and function the right way, they have to have the right sequence of amino acids. And the right sequence of the amino acid is contained inside our genes. So what the genes have is they say, you need to put this amino acid followed by this amino acid followed by this amino acid to build your structure, your protein. Okay? So genes are the cookbook. They have the information how to make proteins. So far clear? Okay. Now it's going to get complicated. So how the process of genes being holding the information is first transcribed. So the cookbook is first copied, and that process is called transcription. It's first copied, and that copy of that gene then leaves the center of the cell and comes outside, and is then used by a machinery to produce the protein. So that translation of the copy of the gene into the product, that process we call translation. So there is a process of transcription, and then there is a process of translation. The translation is where the genetic material is actually translated into your product, into the protein. And just to make you note, DARS2 is a translational disorder. And so we talk about what is the process of translation. Now it's going to get even more complicated. But I have a video, if it works. So here is what I just tried to explain. So here is the genetic information, the DNA string. The DNA string is opened up. There is a copy made of that DNA called the messenger RNA. That's the process of transcription. This transcript leaves the center of the cell, and now this transcript is here inside our cell body, and there is a machinery that recognizes this transcript and starts producing the protein. And I think best is if we actually pull up a video. It's a couple minutes long. And again, you know, I don't anticipate that you guys will understand everything because it's very complicated and I don't understand everything. But I think you get a sense about this process, and it's really key to go through that so that we can explain what the disease is. You need to understand what is normal before we understand what is abnormal.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure. Human cells.
All right. Who does the mic here? Maybe I should turn this off. I think this needs to turn off. Let's see. Oh, here. Okay. Might be also the headsets. No, let's see. That's probably not loud enough. Okay.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus.
So that's the process.
The DNA code determines the order in which the free bases are added to the messenger RNA.
So the messenger RNA is the transcript,
This
the copy of
process
the gene.
is called transcription.
So this is the copy of the recipe. It's now leaving the
Before the
nucleus.
messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA.
Now, that's the process of
The
translation.
ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid.
Okay, I'm going to stop here quickly. So this is key. So as you saw, the messenger RNA came out, was recognized by this machinery that's now going to start making the protein. And the way it's going to make this protein is that genetic information on the transcript, that recipe on the transcript, is recognized by a counterpart of it, and that's called the tRNA. It's this T-like structure. You see, it's like an inverted T. And it recognizes the structure or the sequence because it has 3 DNA bases that have to be mirrored in with whatever is the nuclear information, okay? So the gene, so it's basically making exact copy of the original recipe. You think about it. You have the recipe. You make a copy of the recipe. Now, you have to have a sequence that's a copy of the copy. And that way, you know, if you find, and each of these tRNAs has an amino acid on top, that little ball on top. So a protein is made out of amino acids, multiple amino acids that are stuck on each other. And so the key here is to recognize which amino acid needs to be stuck to which amino acid. And that order is dictated by the sequence. So far clear? Yes? No? Yes? And so let's look at this a little more, because it's got to get a little more complicated.
Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time.
See, it's
As
recognizing
each triplet is read, a transfer RNA delivers the corresponding amino acid.
the amino acids are added on top of each other.
This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
And that's the process of translation in a nutshell, okay? So you have this original transcript that comes out. These tRNAs are extremely important because each sequence on the gene has a corresponding amino acid bound on the tRNA. If it's the wrong tRNA binds on that copy, it will make the wrong protein.
DNA is a molecule made up of 2 strands, twisted around each other in...
So why am I talking about all this? But the reason I'm talking about all this is because when we talk about DARS2, there is an important element to think about. Remember that tRNA that has the amino acids? We have 20 different essential amino acids. And therefore, we need to have 20 different tRNAs. Okay, for each amino acid, we need to have the right tRNA that will recognize the gene. And so how does the body recognize which amino acid has to bind on which tRNA to make that product? And that's done by a group of enzymes called Trna Synthetases. And so DARS2 is one of the Trna Synthetases. So Trna Synthetases are these enzymes here. And these are basically little engines or little, I don't know how to call it, an enzyme. So it's a little machinery where the amino acid is recognized, the right amino acid, and the corresponding tRNA is recognized. And so if it's the right combination of tRNA and amino acid, it will join them together and it will let this go. Okay? So we have 20 different amino acids, therefore we need to have 20 different enzymes that make this combination. Okay? And these enzymes are called amino acid Trna synthetases. So the Trna synthetases are abbreviated as ARS at the end. Okay? So now it's going to get more complicated because we're talking about DARS2. So who still knows what I'm talking about? Okay. 4, 5. If it's more than 50%, we can move on. So to make it more complicated, and that's not us, that's nature, right? Nature decided that it's not enough to have just 20 tRNAs. We need to have also tRNAs that are responsible for mitochondrial proteins. Now, what does that all mean? So these mitochondria are the energy factors inside our cells, right? And so these mitochondria are little organelles that are sitting inside our cells. And these guys used to be their own organism billions of years ago, okay? And so at that time point, they had their own DNA. And so in addition to the 24,000 genes that we have in each cell, in each mitochondria, we also have its own DNA. It's much smaller. It's a little ring. We call that the mitochondrial DNA. It's about 37 genes, right? This is as far as we know. But these 37 genes are important. 13 of them are key proteins and have the information for proteins that are important for energy synthesis in our cells. And so in order for the mitochondria have a thick membrane, as you can see, and so you can't just make the products of the mitochondria inside the cell. You have to make them inside the mitochondria. And so because of that, there are tRNA synthetases that make tRNAs for the cell, and then there are tRNA synthetases that make tRNAs for mitochondria. Okay? And so the way we call them is if they are made for the cell, we just end them as ARS. If they are made for mitochondria, tRNA, they are ended as ARS2. So when we say tRNA synthetase DARS2, that means it's this enzyme that recognizes the amino acid and the tRNA only for mitochondrial protein synthesis. Okay, so that's DARS2. That's what DARS2 stands for. So DARS2 is a gene that has the information to make this enzyme, this machine that in the mitochondria connects the right one amino acid called aspartate with the tRNA. And so DARS2 is a disease where this process is abnormal. So we have a change in the gene, and Becky again will talk about that more later. We have changes in the gene that result in an abnormal activity of this machine that is supposed to make the tRNA. And so now the body becomes deficient of the necessary tRNA, and so when it's trying to make a protein, it's not making the protein well enough. And so as a result of that, the mitochondria do not have sufficient tRNAs to make sufficient protein and function sufficiently. And so there is many changes seen over this gene. I think, are you going to talk about this? I'm going to skip that. So we're going to talk about this a little later today. But basically, this is a disease where you need to have a bad copy in each copy of the genes. So one single copy is not enough.
I wanted to point out, if you had Dr. Vandicknap do your genetic testing, you're probably, before 2014, you're probably on this page.
Yes.
I noticed Ellie's change is actually like the
Right.
one up on the bottom.
Yep.
So yeah, we're all up there.
Yeah.
Use your testing then.
DARS2 patients make some of the protein, just not enough.
That's a key question we've been trying to answer.
Okay.
So
Sorry, I didn't
yeah,
mean to take you off.
yeah, no, and I think this is a very important question. So basically, one of the things that we have found out from the animal work that you're going to see tomorrow is that if you knock out DARS2, if you get rid of that gene altogether, if you remove that gene from your genome, it's not compatible with life. We don't get living mice; they die before they are made, and it's embryologically lethal. And it makes sense. You can't make protein; you're not going to live. So we think that in all patients that are alive, there is no complete loss of this protein. There is either some remnant protein activity, or it's not available only in certain areas. So one of the key questions that we're trying to answer is it that these different changes in the gene result in reduced DARS2 activity, or is it that certain changes will only result in reduced DARS2 activity in certain types of our cells? And it makes it just another layer of complexity. But our working theory right now, which we haven't proven yet, is that most likely this gene is normally functioning despite the mutations in many cells in the body, but only in certain cells, specifically the brain cells, it may be functioning abnormally. That's one of the working hypotheses that Kristin is going to touch that on a little bit tomorrow, but we just don't know the answer yet to that. So don't panic yet. I just want to make sure you're all sort of okay moving forward. What I'm going to do in the next few slides is try to get a little more into the detail here because it's going to dictate treatment. And so we need to understand this really well because it's one thing to just throw something at somebody without knowing what you're doing, and it's another to try to understand first what the mechanisms are and go more targeted into the disease function. So the one question that has been raised is, well, so this is DARS2 is a gene. Again, just to repeat, it's a gene that is responsible for making tRNAs that are responsible for mitochondrial protein synthesis. So it would sort of make sense to think that if you have a problem with this gene, you're not going to have normal mitochondrial protein synthesis, and you may not have normal mitochondrial function. And what goes for that idea is that when you look at this name of the disease, LBSL, the last L is standing for lactate elevation. And lactate is a byproduct of so-called anaerobic metabolism. When your mitochondria don't function well, and glucose is broken down, rather than going into the mitochondria, it produces lactate. And so lactate elevation is always a concern that you're dealing here with a mitochondrial problem. The other thing is that, again, like we said, that we know that if you knock it out, it's lethal, but we also know that if you knock out the gene, what we can do with animals, and Krishna will talk about that tomorrow a lot, is that in the animals, in mice, what we can do is we can get rid of the gene only in certain areas of the body. So there is a study that has been done, for instance, where the gene was eliminated in the heart only. And so these mice developed heart disease and died. And so when you look at those heart cells, you do see that there is abnormal mitochondrial function there when the gene is completely gone. But again, the challenge that we've had is it really gone in the patients or not? And that's the answer that we're trying to pursue with some stem cell technology that we'll talk about tomorrow. The problem is that, and this is right from the beginning, an original description of Dr. Van Dyck, now back in 2002 when we first met, she presented to me the data, said there's no mitochondrial dysfunction. She had looked at muscle cells and skin cells of the patients, and this has been done by multiple groups. And when you look at the skin and the muscle in these patients, you don't see any problem with the mitochondria. And so that comes back again to the question, is it that the mitochondrial dysfunction is too subtle to be seen, or is it more pronounced specifically in the brain because this is a more specific defect that affects really the brain function? There's another issue that brings it in, and so this is really the key issue right now. This is really complicated. I'm still only understand of it half myself, okay? So don't get offended if you don't understand this at all. But I just want to introduce here an idea that I think is really important in this disease. So there is a pathway called the integrated stress response. This was first described in 2003. So it's a novel concept, was not really known in biology or in nature, in neuroscience or in natural processes. But it turns out to be a really old system that has been around for billions of years. So throughout the process of evolution, first there were bacteria before we had the big cells, right? And so even in living bacteria that are billions of years ago, one of the things that bacteria did to protect themselves was having this integrated stress response. So what does that mean? If there is any stressor, so if there is starvation, lack of glucose, if there is an infection, if there is oxidative stress, if there is any other form of stress like iron deprivation and so on, these things turn on different molecules. And you see here these 4 master regulators, these molecules here. And they give the cell the order to shut down. It's sort of a hibernation protection strategy. So the bacteria are starving, and they can either die because they're starving, or they can shut down their protein synthesis just to calm down until the starvation period is the stress is over, and then they can turn back on again. So it's a protective mechanism that is in place billions of years, and then we have that in every single cell in our body. And so this integrated stress response is directly related to the process of translation. Remember, translation means taking the genetic information and making the protein. And we said DARS2 affects this issue, right? And so it turns out that there are multiple molecules that regulate how much protein we can make based on the amount of stress that we have. In no stress state, we make a lot of protein. In a high stress state, we make very little protein. That's just how the cell is regulated. And if you have a problem in the process of translation, the hypothesis is that as a result of that, you get a dysregulation of this process, and you may have too much of a stress response. So an exaggerated stress response results in a lack of recovery. And so in a clinical setting, what that would translate to is that if you have a stress, you have a viral infection, you have dehydration, you turn on this integrated stress response. In a normal person, there would be a quick recovery. But here, there's this exaggerated stress response, and so it takes longer to recover. And then, any of you who have a child with this disease know that this is sort of what you see clinically often. And so, in fact, when you look at the mouse models, and this has been done so far in the heart, and we are now chasing this down in the stem cells. We don't have the answer yet clearly, but it looks like when you look at this pathway here, when you have decreased DARS2, so when the gene is affected, you have a mutation, or you knock out the gene in the mouse, in heart cells, you get an upregulation of this integrated stress response, and so you go into the so-called hyperprotective state, a state where you're just hyper-responsive to the stress, and so it takes you longer to recover. So now we can come up with two hypotheses why you have disease when you have mutations in this gene. One is the initial hypothesis that there is decreased DARS2, so there is decreased mitochondrial protein production, and there's a decrease of mitochondrial function. And so you get oxidative stress as a result, you get inflammation as a result. And looking at the animals, we see oxidative stress and we see inflammation in the brain. And again, Christian will talk about that. So we do think that this hypothesis is true, that there is mitochondrial dysfunction. But we also think that there is a second mechanism where this disease is affecting it. And it's not uncommon at all in nature to see that. Usually our natural systems and bodies and cells and anything that we have are quite smarter than we actually are. The body protects itself in many ways. And so usually just having one system shut down is not sufficient to cause disease. You usually need to have multiple systems to shut down, and then the entire system collapses. And so the second hypothesis that for which we are starting to have data is that this abnormal DARS2 results in this dysregulation of this integrated stress response, and therefore a hypersuppression of protein production and sort of this delayed recovery. Now, there are people who will say, no, the first one is true, and there are people who will say, no, it's not, it's only the second hypothesis. Our thinking is we are diplomats, we think both hypotheses are true. And again, it makes more sense in a natural system than you need to have multiple things falling apart before you actually get sick. And so this is key here, because when we move forward towards therapeutics, if we focus only on one aspect, it's probably not going to be sufficient. And so we need to understand this more, we need to study this more, and we are doing that right now. And we may have to target both issues here to be able to get the patients actually to stop progressing and improving. So I have just a few things that I want to talk about. I don't know whether we are running. Actually, we are good in time because Dr. Werner is 11:30, I think, or 15, her talk.
She's 11:20.
20, okay.
And you have about 10 minutes.
Okay. Mm-hmm. Go ahead.
So are you saying that the DARS2 dysfunction may not be in all the cells of the body? It might be more stuck in the brain area?
Yeah.
Okay.
So there's two theories there. One, the more energy you need, the more mitochondria you need, the more likely you're going to be affected if you have a problem, right? And so guess what? Which cells in our body need the most energy? It's our brain, right? It's the heart second, and it's the gut third. And so many times, often in mitochondrial diseases, you will have the brain, the heart, and the GI tract that are very vulnerable to things. The other issue is, though, more complicated. And the other issue is that, and this is really complicated, but I don't know, are you going to, Becky, talk about the splice site and all that? Or yeah, so you'll hear it in the afternoon. I think it's just, I want to just minimize the information exposure in one talk. But there is a problem with the genetic mutation that may be selective to the brain, meaning that the splicing, abnormal splicing may be normal in certain parts of the body, but not normal in the central nervous system. But that we are still trying to understand. So I wanted us to talk a little more about the clinical things, the few things that we have learned, and the many things that we don't know yet. So one of the things that we have learned is that we've seen now some adult patients, at least in the literature, that are getting into their 30s and 40s, and so there's data accumulating over decades on these individuals. And looking at them, it looks like clinically people are getting slowly worse, very slowly again. But and there is always these episodes of sort of spontaneous recovery after they have an acute illness. But when we look at the MRI, it's interesting that it looks like it's actually getting better. And so the question is, what do we mean when the MRI is getting better? And so when you look here, this is an individual, this is from a paper, nobody who's here today, whose MRI looked like this at 10 years. You see all the white stuff is abnormal. Then at 18 years, this is how he looked like. And then at 22 years, there's still abnormal white matter changes, but most of that swelling and the bright white matter is actually not there anymore. And it's sort of better. One of the things that you will notice here is that there is a lot of swelling here and that there is a little bit of shrinkage there. So what we see here, it looks like that there is sort of this swelling in the brain during early childhood. And then the MRI seems to get better because the swelling kind of calms down, but those areas of the brain that were swollen are also partially no longer there, and as a result of that, there's a little bit of a shrinkage. And so we don't exactly know what this means, because the good news is this is a mild disease, and so most people are still alive, and so we don't have, the bad news is that as a result of that, we don't have any brain autopsy material really available. There were 2 individuals who were autopsied that were babies that passed away in a very severe form of the disease, and we can't probably correlate that with most patients who are alive because they were a lot more severely affected. But what we're thinking is, we see in the animal model, and Christina will get into that, we see inflammation in the brain. And so one of the thoughts that we have is that there is this initial period of inflammation that happens that swells up the white matter and gives you this bright signal, and that it looks like even in the mice that are clinically getting worse over time, this inflammation kind of calms down. It's like, you have an acute attack and you're being attacked, everything is swollen up, and then things kind of calm down over time, but you had casualties. And so you see this remnant left over, but we think that as a result of this, the MRI may actually not be necessarily a helpful measure to predict if the disease is getting worse. Okay? And so that's key, because we need now to think about, okay, what are we going to be looking at if we're going to do a drug trial? So there's a lot of things we don't know. The very first thing is, does the change in the gene actually affect how severe the disease is and when the onset of the disease is? That's something that's been looked at a little bit, but we are hoping to get the data of any person we can catch through our internet, basically in the study that we're now starting, and you'll be contacted if you haven't yet. And basically collect this information to get a better understanding of, are there mutations that are more selectively causing more severe disease? This hypothesis has been raised by many, but there's some counteractive things. There's a case report of 2 siblings, one of them very severely affected, one of them very mildly affected, and they both had the same mutation. So we still don't know whether the change in the gene necessarily correlates with disease severity. The other thing that we really don't know is yet how, you saw that plot of how many people need assistance walking, but we really don't know, how does this disease get worse, or does it get worse? Are there individuals who are completely stable over a lifetime? Are there individuals who are stable and then crash at some point? So this is all things that we really need to study before we can say, oh, we're treating somebody, we want to see if you're getting better. Well, what is getting better? Is it we know that in these episodes, people decline and they have recovery. It may not be 100% recovery, but 90% or 95% recovery. And so we don't know how the MRI is going to change, and we don't have yet any marker that we can look at that allows us to predict. Imagine a world where you're born, you have this disease, we do a blood test and say, we predict that at 5 years you will have this. And so now we're going to start treatment, and then we will see if we actually had an effect. We don't have that sort of what we call a biomarker yet. We also don't really know exactly what are the risks for the triggers. We hear obviously the reports from families that dehydration, infection, head trauma are triggers for induction of episodes, but we haven't really looked at it systematically, and we would like to do that. And that's an easy thing to do, and I think you guys can help to get that information done very quickly. And then again, the mechanism is something that we have to figure out sort of in the lab. So I'm just going to finish up in the last couple minutes. I'm not sure if I should. Is Dr. Vernon here? Oh, you are? Great. So let me try to wrap this up, because this is really Yeah, let me just finish up with this one slide and skip some of the other stuff. So when we talk about treating a genetic disorder, and this is sort of generic for any genetic disorder, there's several therapeutic strategies to think about. So you have a gene defect that results in an abnormal product, your protein, this DARS2 enzyme, right? And that results in a white matter disease. And so there are different interventions that you can do at different time points. You know, the sort of the no-brainer thing is, well, can we fix the gene? You know, can we go and change the sequence of the gene? That is a technology that is being developed, and it's extremely difficult to do right now. It's this CRISPR-Cas technology. You can basically go in and cut out a gene and replace it with the correct sequence, but there's no feasible way right now to be able to deliver that construct that does that, the machinery, into the brain, and that's being investigated. And I think we are somewhat far away. The alternative thing is that a lot of people, what they do is they just infect the person with a virus that has the correct sequence. So we say, okay, you have this abnormal gene that's not doing the right product, but we're going to give you a virus. It's like the Matt Damon movie, I forgot what it's called, Bourne something, you know, Jason Bourne, yeah, that's what they happen to them, right? So they get infected with a virus, they get a viral infection, affects the nervous system. It has to be a virus that has a high tropism for the brain. And basically the gene is then brought into the brain, that's the correct gene. And so it will integrate itself into the cells, and so that way you basically are overcoming the problem. The main challenge with the gene, that's what we call gene therapy. And usually we use viruses, and this is already hitting clinical trials. It's already happening. It's nothing futuristic. It's actually, there is a trial right now going on at NIH for a very similar disorder that affects predominantly the spinal cord and motor neurons called SMA, where now they're getting positive results from the gene therapy trial. The problem is getting it to the brain is much harder than getting it into the spinal cord. And so there are different ways to do that. You can inject it directly into the brain, but then we see that it doesn't spread to the other side. You can inject it into the vein. And so one of the things that we are pursuing here, hopefully in the next few months to year, is an intra-arterial delivery where we can inject things into the artery and see if we can get it directly into the brain. So then the other methods are, rather than gene therapy itself, can be well, let's go back to the ultimate. Can we restore things? Can we give people stem cells that will go in and make up for the damaged myelin sheath and the damaged nerve cells? Theoretically, yes. We're doing this in animal models. Clinically, it's extremely difficult to do. And the reason it's difficult to do is when you inject the cells, it's very hard for them to get to the place that they need to go to integrate and to take over the function. There was a trial that was done a few years ago by a company where they went into a disease called Pelizaeus-Merzbacher disease, another leukodystrophy where there's no myelin. And they injected these neural stem cells in 4 different spots in the brain. So it was brain surgery, basically, with the hope that they're going to integrate, become myelin-producing cells. And while the result of this study was that it was a safe procedure, they didn't really have any clear beneficial effects. And the company ended up going bankrupt. And the reason it went bankrupt was making those stem cells. Well, guess where they came from? They came from aborted fetuses. And in order to scale this up for a clinical trial, you just need a lot of cells and you need a lot of abortions. And so that just doesn't make sense, and it didn't really pursue. So the last strategy is now to try to use molecules that just go in and fix these defects, right? The mitochondrial defect that we talked about, the integrated stress response that we talked about. So those are the strategies that we are pursuing right now in the lab. I have to say that I can't give you much of a detail, but there's a couple companies that are very interested in this integrated stress response and have contacted us because they're interested now in LBSL. And so think about it. You know, we have a handful of patients here. There's already a company that's actually interested because they have a drug that will work against the ISR aspect. So there's I think a lot of optimism there. If we get good results in the animals and if we can get that hint towards that direction, then we might be able to come up at some point with a clinical trial. Well, before we get there, we need to collect all these unknowns because if I take a package to the FDA, and my better half works at the FDA, so I know it too well. But if you go there, they want to know. Okay, they don't care that it's a rare disease. There are trials being approved for drugs with 11 patients, 12 patients. Dr. Vernon is doing a trial. How many patients do you have in your trial?
12.
12 patients, right? So you can do mini trials in ultra-rare diseases, and the FDA is fine with that. But you have to know exactly what you're going to be looking at. You can't just say, is the patient getting better or not? You have to have quantitative variables that are not only quantitative, but also meaningful to the patient. So if you have a laboratory value that improves, that might look great on the lab, but if the patient didn't improve, it doesn't matter. And if the patient doesn't feel like he has improved, it doesn't matter at all. So, we come up with things like, we went to the FDA for another disease. We proposed that we're going to measure how much you sway when you stand on the test. And we thought that's a great measure of balance. And the FDA is like, we don't care. Does it matter to the patient? You have to correlate that degree of sway with the quality of life of the patients. If you think that if the patient said, we don't care how much I sway, then you can get an improvement in sway. That doesn't tell us anything. So I'm going to stop here because we have a great speaker following up. So I just want to thank, again, the Cure for LE Foundation. Every single piece of research related to LBSL done at the Moser Center was done because of the funding that you guys have raised, and it wouldn't have happened without it. We didn't have a program before that. There's multiple people in this room that have significantly contributed financially to this, and I really appreciate all you've done. We need more, but I think that we are hopeful that we can get some data very soon that we can go and pitch to national funding.